Chapter 8, Section 6. Regulation of endospore formation. You have to remember, endospore formation is going to be in response to adverse or some extracellular signal that triggers this response. A vegetative cell is being a vegetative cell, doing all the stuff we've talked about in the previous 5 sections until something changes. And then it's going to trigger this endospore formation. Once whatever that environmental stimulus was, lack of this, over accumulation of that, once that changes, then germination will occur. The endospore will open and out comes a vegetative cell again, which will start up all the stuff we've talked about in sections 1 through 5. So when you look at what's occurring, what we see here in this picture is that as the spore is forming, this pre-spore, the mother spore right here, the mother cell, is becoming depleted because all of its nutrients are going here to make this. All the nutrients, all of its amino acids, all of its nucleotides are going to making the new chromosome. All the proteins, the polypeptides are being shuttled here. They're going to be needed. White, yes, right here is the spectrum. White, highly rigid, black, extremely elastic. So what we see here is the cell wall is being remodeled. Everything going on in here is basically depleting. Everything's being shuttled here to be used to make all of those different layers of the spore. As I said, some adverse stimulus, external condition, external signal, starvation, lack of nutrients, certain nutrients are now depleted. Water's going away. Temperatures, too high, too low, something. There are different sensor kinases, different proteins that are kinases that are embedded in the cytoplasmic membrane. That they're acting as a two-component regulatory system. We talked about these in Chapter 7. The membrane bound gets triggered, and then it passes on, a phosphorylates, an internal protein in the cytoplasm, which starts a series of pre-programmed cellular responses. Activation of certain gene transcription, turn off other genes, activates certain metabolic pathways, turns off, inhibits other metabolic pathways. Well, what we see here for the ones that are responsible for endospore formation, these kinases, they ultimately end in what is called the sporulation factors. It's a series of proteins in the cytoplasm that they only get triggered right before a sporulation event. Well, these will then go and regulate different operons, regulons, whatever. And one of the proteins that's mostly responsible for this is SPO-OA, sporulation OA. This is the one, once this is triggered, once this has been activated, phosphorylated, sporulation occurs. So it's not just a certain one of these sensor kinases gets tripped and it activates its component in the cytoplasm and then boom, everything goes. No, it's a slow accumulation of more and more of these kinases get tripped. More and more of their cytoplasmic second component gets tripped. More sporulation factors get tripped. More of the SPO-OA gets phosphorylated until it hits a certain level, and then sporulation will occur. Some external signal. You have SPO to AA, SPO-OA, SPO to E. Some external signal triggers accumulation of SPO-OA to be phosphorylated. SPO-OA goes and removes the phosphate group, or activates, SPO-2E, which then goes and dephosphorylates SPO-2A. Okay, SPO-2A activated goes and plays well with SPO-2B. SPO-2B goes and activates a couple of different sigma factors. So SPO-2A pulls off 2B, which allows sigma factor F to be activated. Boom. Sigma factor F triggers a whole host of responses. Sigma F goes, activates sigma E. Sigma E spreads throughout the cell and starts triggering all of those different layers to be laid down. And it's just this domino. Do I expect you to know, E before, F before E before K and G, and no. That's stuff you can look up. Two components, kinase, regulatory kinase, receptor systems, signal systems, however you want to say it, think about it. SPO-A gets activated. SPO-A goes and activates 2E. 2E dephosphorylates SPO-2A. 2A goes and pulls 2AB off of sigma F, releasing sigma F, which can now go and start the process of activating the different sigma factors. The sigma factors which are going to control expression of different operons and different regulons. Those genes from those operons, from those regulons, are going to call for the making of the different layers. As you see here, F and G make the 4-spore. 4-spore, remember, this is when certain layers are being laid down. We're starting to get the new copy of the chromosome and some of the ribosomes and stuff shoved into the area that will become the endospore. E and K, sigma F, E, sigma K, are going to activate certain genes in the mother cell that are going to make, remember, this is the regulons and the operons that are going to make the proteins that become the enzymes that start laying down different coats, different layers in the endospore. And then when you go and you look at this, SPO-A activates F, F activates G and E, G goes and does E, E goes and does K, G and K go and do this, and blah, blah, blah. No. If I ask in a test question, hey, sit there and tell me about the regulation of, yeah, you can go and look this up. Who does what to whom? But to sit there and know this and memorize it, no, that's asinine. What's interesting is not all cells that sporulate are going to sporulate at the same time. Turns out you take Bacillus subtilis is a great example of this. Bacillus subtilis is one of these endospore formers. The first ones that to be triggered to activate SPO-OA, which means they're now well on their way to forming an endospore, will actually produce toxic proteins. That's part of those operons and regulons that are now being upregulated, being activated. These toxic proteins will then be pumped out into the environment to cause other cells to lyse. Remember, all cells are basically just bags of amino acids, sugars, lipids, and all sorts of good stuff, which that Bacillus subtilis that got the toxins out first gets to now transport in. Why go and make all this stuff when you can kill your neighbor and steal it from him? Literally, that's Mother Nature at her finest. As these toxins are being released, well, it turns out some of the things that are also being released, transported out, excreted out, are proteins that will delay sporulation in neighboring cells. So it's not just, it's not, hey, look, let's all do this together. It's still every cell for itself, even though they're the same species. And not only am I going to slow down your ability to do the sporulation and protect yourself, but I'm going to try to kill you to get out all of the goodies inside of you. Once they hit a certain point, many in this event, cells can start making an antitoxin that'll protect them from this toxin, but they got to get a certain point into this sporulation event. If it's still just a vegetative cell and hasn't started really responding yet, it's completely, going to, how do I say this? It is completely susceptible to the toxins. It will be killed. But if it's far enough into the sporulation event, it's now making an antitoxin, and it's still in the race to who can get to form an endospore and be protective first.